Hello, this is the Tuesday activity replicating DNA, and it's a clever worksheet. And what I'm going to share with you is just a couple of tips here. First off, this group of cutouts up here are for you to uh, copy and paste down on this backbone. This little white box off of the thymine is showing that this is a very strong covalent bond. So this bond should attach to the backbone. Now, I want you to go back and double check. It only attaches to one or the other. You'll have to go back and look at your notes. Should it be attached to the phosphate group or the sugar? And what you're going to do is you're going to grab it, pull it down here, make sure that you've got the bond side rotated so that it clicks right on the proper spot. Now, you are asked to just create your own segment of DNA. So you go ahead and write in A's, T's, G's, and C's here that you think you want to make for your own personalized copy of DNA. And then you want to add in the complementing base pairs following Chargaff's rule. You want to go A to T, G to C. So if you have an A here, you're going to put a T here. If you have a C here, you're going to put a G here, and so on. So fill those out. Now, this is going to be your original strand of DNA to work with as you build a fun model down below here. So let's go to part 2. Now that you've created your original strand, go back up and grab these little parts up at the top and bring them down. So go up and grab, if you started with a T here, grab a T, bring it down and connect it here. If it was supposed to be matched up with an A, grab the A, bring it down and put it right here where it's supposed to be matched up, and so on. So if you had a C there, put your G here and just add them all on. Make sure that you have all of those complementary pairs. Now, as you notice, when you get a little farther over here, you're not going to be able to be touching each other. That's because you're building a model representing that these two pieces have zipped apart. And they've zipped apart because the helicase is coming right down the middle. And what it's doing is it's creating what's called a replication fork. So grab this helicase and put it where you think it would be in the unzipping of the original strands process. Then you are going to put in 2 DNA polymerases. And then when you have a little more space out over here, have those polymerases adding in a couple of the new pieces to create the 2 new complementary parts for the original strands. So you're just trying to model the process of replication. Over here, replication hasn't started. Right in through here, you're going to have your helicase unzipping. And then here, you're going to show that DNA polymerase bringing in parts. And all you're going to do is just reuse these cut and paste parts up here. When you are done, answer these questions. And they're pretty straightforward. I really like the question at the end here. It's asking you to think about photocopying information and how is photocopying similar to DNA replication and how is it different? All right, good luck with this assignment.